Hello, everyone. Welcome to another webinar from GenomeArc. And we have a very exciting talk today, and we have a very exciting speaker. It's our very own Mariam Eldesouky, who is the Science Communication and Marketing Officer at GenomeArc. And she will talk about from data to diagnosis, automating genomic tertiary analysis for rare disease. So she will talk about in depth about rare disease genetics and how the complexity arises for diagnostics and therapeutics, and how those issues can be resolved with our GenomeArc solution, GenomeArc Horizon platform.
So thank you for the introduction, and it's a pleasure to be here today. My name is Mariam. I'm going to be talking about from data to diagnosis, automating genomic tertiary analysis for rare diseases. I think it's a really important topic that intersects with many different crucial topics. So there's genomics, there's how to interpret data, and then there's how to apply to different diseases. So let me start with the main disease perspective that we're going to be talking from. That would be rare diseases. And rare diseases are 80% genetic in cause. They affect more children than adults, and the average timing of diagnosis is about 5 years. So you can understand how long and how painful the process is for the patients. And this is just one of the diseases that is related to genetics and is also needed for diagnosis and treatment. And so we come to a concept here, which is diagnostic odyssey. So diagnostic odyssey is the process in which the patient goes from doctor to doctor and can't find the reason for their disease. They can't start the treatment process because they have not been diagnosed yet. And then when we go to the extreme cases, the extreme end of rare diseases, we see NICU and PICU cases. So we have neonatal intensive care unit patients, and we have prenatal intensive care unit patients. And they have a higher fatality rate compared to most rare genetic disorders. A large portion of them carry lethal mutations. And then we need to think about how automating the process of genetics, how to get a genetic diagnosis faster can impact the lives of rare diseases and especially these critical case patients. So when we talk about the impact of automation, I want you to keep this in mind. It impacts, especially in these extreme cases, a faster diagnosis turnaround time, hours compared to weeks, and then accuracy, much higher accuracy, and then impacting treatment management. It has a direct impact on how the patient is treated, especially in NICU and PICU. And so we're considering here two factors. We're considering genomics, and then we're considering the time impact on patient care. And so now that I've just given a brief background about the diseases, the rare diseases that we're going to talk in context of through this talk, let's move on towards genomics. So right now there's a shift towards precision medicine due to two things. There are multiple factors. However, two of the main factors are sequencing availability and reduction of cost. So we have whole genome sequencing, whole exome sequencing, and then the cost of sequencing has decreased, as you can see here in this graph. And so what we need to understand here is that there's been a shift in the entire world towards genomics and then towards a higher picture, which is precision medicine. So there are some emerging technology as well, like whole genome and whole exome and long-read sequencing that have encouraged this shift. And then we also have precision medicine, the precision medicine concept, which combines genomics with other data to achieve a personalized approach that is evidence-based. So right now we have moved on from looking at the DNA towards how much impact can we create through understanding our DNA, and then tailoring this in healthcare for each individual. So then when we look at genomics, we need to understand that there are multiple levels of analysis that happen. So we have primary analysis, secondary analysis, and tertiary analysis. So primary analysis is just the sequencing of the genome or exome, or even a targeted gene panel. And then we generate the raw reads. And then after that, we take these reads and we align them to a reference genome. This here happens in the secondary analysis. So in the first analysis, in the primary analysis, we get just raw data that make not a lot of sense until we process them further. And then we have secondary analysis where we align them to a reference genome and we produce a BAM file. And then from that BAM file, we call variants. We identify different variants. Still, we have not identified their impact yet on the patient or the phenotype. However, we've just identified what variants are there. And then we get from that a VCF file. So until now, we're still in the secondary analysis. Now, once we start determining which of these variants are actually impacting the patient or impacting the phenotype or the disease pathology, then we have moved on to the tertiary analysis. So in the tertiary analysis, there is the traditional workflow where there's manual processing and interpreting data. It's a complex and lengthy process. And then we get to the report. Or there are emerging workflows supported by AI or machine learning to actually automate this process and make it faster and easier. And so when we talk about tertiary analysis, so we'll be focusing on tertiary analysis in this talk, because that's the part where we really connect genomic data to all further applications. So until the secondary analysis, we've just really, so until here, we've just processed the data. But then here, we're starting to connect the data to other omics. We're starting to connect it to a report, which can impact a patient's life. So it's really where genomics impact can be seen the most. So when we discuss about tertiary analysis, we need to consider, of course, ACMG guidelines. So the American College of Medical Genetics, ACMG, have developed guidelines to help in the interpretation of data. Now, these guidelines depend on different evidence-based criteria that support the pathogenicity of a variant. So if you look at this table here, this is for SNVs, single nucleotide variations, and indels. These range from strong to very strong, but in two different directions. We have pathogenic and then benign. And then you have these different separate columns of different information regarding the variant. So you have whether it's prevalent in the population, whether there is computational data that predict the deleteriousness of the variant, if it's de novo or not, the inheritance, functional data. So there are different criteria. And then based on these criteria, you can stack the evidence whether and decide whether the SMV or indel is pathogenic, likely pathogenic, VUS, benign, likely benign. And so we usually use different bioinformatic tools and databases to provide these evidence for each genetic variant. Now, right now, this table and all these evidences are just for SMVs and indels. But then if we move on to structural variants, like CMVs, insertions, inversions. So we have for CMVs here, copy number variations, there are different guidelines. This is a table taken from, again, the ACMG. So they have two separate papers. I invite you to look at them. There's one for SMVs, the previous table, and then this one is from their CMV paper. So here, these guidelines have some differences between copy number loss, where the variant is a deletion, and then another table for copy number gain here, which is for duplications. You can see that there are different sub-criteria for each criteria, right? So we have, this is overlap with established predicted haploinsufficiency or benign genes or genomic regions. So, and then you have different cases in how it overlaps and what it affects that determine how much you add or subtract different scores. So you can see that the process can get a lot more complex when you're evaluating not just SMVs, but also structural variants. And then another added complexity is the fact that there aren't a lot of databases and/or tools available to evaluate structural variants. So there are a lot of tools that can predict deleteriousness for SMVs, but then when you come to structural variants, it's a little bit more hard to find tools for that. And so we arrive at the bottleneck of genomics. So right now we have started having sequencing is streamlined, primary analysis is streamlined, secondary analysis is streamlined. But then when you come to tertiary analysis and you want to determine the impact of variants, whole genome sequencing provides how many variants? 5 million on average. So you want to determine which of these 5 million, maybe 10 or 20, that might be impacting the phenotype or disease. So you have a complexity in the interpretation of the variants. And then the second thing is because there is this complexity and inability to streamline the process currently, there's of course an inability to connect the genomic data with other omics in a scalable and efficient way. So first one is interpretation of the data, and second one is how we integrate it with other omics in a scalable way. And so we need three factors for that. We have timing, we have accuracy, and we have integration. So we need to combine these together, and that's what we did with our platform Horizon. So GenomeArc Horizon is an ultra-fast precision genomic platform that is really amazing. It streamlines the entire process and then connects the data. So onwards from this point, I'll be telling you about how we can do tertiary analysis and how we look at the variants, how we prioritize them, as well as different cases, and then how Horizon helps with each of these factors as well. So these are the outline, these are the points I'm going to be discussing further on from here. So the first one is how to connect phenotype to genotype. Second one is ACMG-guided variant prioritization or classification. So how we classify the variants based on ACMG guidelines. And then the third is family analysis, where we look at inherited variants versus de novo variants. Fourth one is preventive carrier screening and secondary findings. Here we are moving closer towards the idea of personalized medicine, precision medicine, and how we screen for carrier screening and findings that are not primary. And then finally, 5, pharmacogenomics and multi-omics integration. So for the first point, phenotype to genotype. So we have something called the Horizon Phenogene module. We have a large language model that processes clinical symptoms and provides three things: HPO terms, probable causal diseases, and the gene panel of potential implicated genes. So here the main thing is that we made a survey before, and a lot of people have mentioned that one of the main things that complicates the variant interpretation process is the lack or inability to connect the clinical indications in the patient with the genomics or with the variant interpretation process. So this part especially gets complex when we're dealing, as I mentioned, with rare diseases where sometimes there's no diagnosis. There's just a list of symptoms. So here, this can help in three things. Number one, no more diagnostic odyssey, or at least we can reduce and help in it. Number two, we connect the phenotype to the genotype, so we have a focused analysis. And number three, there's a starting point now for phenotype-based genomic analysis because the large language model, as I said, provides three things: HPO terms, probable causal diseases based on the symptoms that you input, and then the gene panel for these potential implicated genes. So let's have a look here. This is some of the output from the phenogene module. So we have here, for example, So this is the unstructured clinical phenotype written by the user. So we just write some symptoms. And then based on that, Horizon outputs HPO terms, and then it outputs this graph, which is genes that might be implicated in it. It also outputs another graph, which is probable causal diseases that might be causing these symptoms. But then if you look at this graph, we have a curated gene panel just from a list of symptoms. And this is based on millions of training data points. So the first part of variant interpretation, which Horizon helps with, and which should be done when we're talking about tertiary analysis, is doing a phenotype-based analysis and sort of curating a gene panel. So this is one of the benefits of automation. And then we move on to ACMG-guided variant prioritization or classification. So there are three things we need to combine here in this step, and this is the most important step, where we find which variants are actually impacting the phenotype or disease. So the first one is we implement ACMG guidelines. We follow these criteria that were in the tables for SMVs and for SVs. And then we do variant prioritization. We filter out based on these criteria and we prioritize certain variants. And then finally, we analyze from these prioritized variants for SMVs and SVs, which ones are impactful, which ones are clinically significant, and are pathogenic, likely pathogenic, VUS, or maybe likely benign or benign. So when we talk about variant prioritization, there are certain criteria that are based, of course, on ACMG that we do in the beginning to sort of minimize or prioritize certain variants. So when we talk about whole genome sequencing, we usually get a VCF file that contains on average 5 million variants, which is a huge number to do manually. And then if we're talking about exome sequencing, we get around 100 to 200,000 variants. So again, this is a huge number to look at in SMVs and SVs. So what we do is first we filter for rare variants. This can be a cutoff from the frequency. Sometimes it can be 0.005, or sometimes it can be 0.001 even. So we prioritize and we look only at rare variants. And then the second thing is that we prioritize variants that have ClinVar information. So we prioritize variants that have ClinVar information, and these are previous reports on the variant. And then the third thing is we search in proprietary databases for the variant, if it has been there or not. Finally, we look at bioinformatic tool scores. So we first filter for which one of them is rare, which ones are rare, and then we look at ClinVar information to determine whether the variant has been reported previously, which is a very important evidence that it might be related in the pathology of a certain disease. And then when you look at databases or when you look at previous reports, sometimes you might need to check actually more than ClinVar sometimes, which is perform a literature search or in specific databases. Now, this is where having proprietary databases, such as the ones in Horizon, can help you in having already curated data sets from literature, which speeds up a lot of time and effort. So you'll find that the variants reported in specific papers in a specific phenotype with a specific cohort are already there in a table, and you can just check where they were found, and then you can go to the paper immediately rather than performing the literature search. Finally, you need to also predict the deleteriousness of the variant, which we do using bioinformatic tools that would be like CAD, Revel, PLI. So these are all integrated into Horizon, all of these information. So then let's look at identifying, this is a case using Horizon. Let's look at how we identify here the pathogenicity of variants, which is ACMG guided. So each SMV has this interpretation here. For example, this one is likely pathogenic. It's a variant in the titin gene. So it's interpreted based on ACMG in an automated way. Imagine you have a list of variants, 10, 15 variants that have this pathogenic or likely pathogenic interpretation. Then you have a whole list of VUS variants, and then the rest are benign or likely benign. So this analysis time takes around 10 to 20 minutes maximum for approximately 5 million variants from whole genome sequencing. And then for whole exome sequencing, you get it's done in 5 minutes. So 5 minutes, you already have the variants prioritized, and you can sort of look at a filtered list where you can examine the evidence for each variant based on ACMG. Now, there are 3 databases that are queried. You can see here, these are the basic variant details. Then you have 2 tabs, clinical information and omics. And then you have 3 databases that are queried for this. So you have proprietary databases and public research databases, and then custom databases. So when you look at variants, the main questions you need to ask is, has this variant impacted or has it been reported in previous cases where it's impacting the patient or the case with the same phenotype, for example? In that case, this would be in proprietary databases that are derived from literature and from other research collaborations and are unique to Horizon, for example, or if you do a literature search yourself. So that's one way. And then the second thing is the variant rare? That would be looking at NOMAD. Is the variant predicted to be damaging? So that would be looking at the scores of CAD, REVEL, or PLI. And then another question to ask, another important question is it predicted to be damaging? Again, is the gene pathogenic? So in that case, you would need to look at the pathogenicity of the gene. Has it been reported to be implicated in different disease mechanisms, maybe through GWAS studies or through functional studies? And then another important thing is to consider your own custom data. So that's another thing that is integrated into Horizon's databases, custom databases. So for example, let's imagine that we're looking at a specific population. So you need to consider population-specific data towards this variant. And of course, it's known that there isn't a lot of genomic diversity currently because most data is derived from European ancestry data. So then sometimes it is not representing accurately different populations. So it's important that when you have, for example, a custom frequency database for a certain population, you will have the ability to integrate that and check in the analysis whether it is rare in this specific population or has it been reported in your cohort. And then when you go to Omics tab, you also have this transcriptome. So we're expanding a lot on the Omics tab, and I'll be showing you a sneak peek at the end of this talk about the features that we'll be adding. So in this way, the main thing is to get evidence based on ACMG that shows you whether this variant is pathogenic or not. And Horizon automates the process by giving you the interpretation in 5 minutes for exome, 20 minutes for genome, whole genome, and then it gives you all this evidence, all in the same place, so you can look at it and validate yourself. Now let's go to SV variants, structural variants, which are much more complex and they don't have a lot of databases or tools that predict that can help in predicting pathogenicity. So again, Horizon displays all the information supporting the interpretation, and it's based on ACMG guidelines. So when we look at a structural variant, we're talking about, for example, in this case, we're talking about CMVs specifically, copy number variations. So we're talking about a long stretch of DNA that has impacted, that might have impacted multiple genes. It might have inside it, it has impacted protein coding regions, or it has taken a large chunk of the genome, basically, a large chunk of a specific part of the genome. And so, first of all, we need to filter for whether it is prevalent in the population or not. Basically, is it a rare CMV or not? So for that, there isn't currently like a controlled population frequency database for SVs. So we have an integrated one in Horizon where it has a lot of data points of established control SVs. And so Horizon evaluates the structural variant on these control SVs and checks if it's already in the control SV database. And then the second thing is um, finding if the SV has been reported in previous cases or not. So ACMG guidelines have multiple criteria for the SV. The main ones are this. First of all, it should not be reported in an established benign region, or it should be reported if you're classifying it as pathogenic. It should be reported in previously reported cases or pathogenic regions. That's number one. So previous reports are important. Number two is which genomic region or has it impacted, or has it impacted protein coding regions? So that's another important point. What is the genomic content that has been impacted by the CMV? Number three is how is the number of genes, how many genes have been impacted? And then are these genes dosage sensitive? Are they haploinsufficient or triplosensitive genes? So then previous reports, genomic content, and the size of the CMV as well. So you need to consider all of these when you're looking at a structural variant. So then when we talk about proprietary databases that are included in Horizon, so we've talked about the control database. So then we move on to the proprietary databases. Is the structural variant overlapping or matching with pathogenic SVs that have been reported in previous cases or databases? So that's a very important part to look at. If has this SV overlapped with other reported structural variants? And then the third thing is the genomic content of the structural variant. What genes are impacted by the SV, and are these genes pathogenic or have they been reported in different phenotypes? So if we look here, this is from Horizon platform. You can see here, this is a CMV. We have the basic variant details, chromosome start end, length, variant type. And then you have here the impacted genes shown on the chromosome browser. So you have here, this is the impacted gene P10. And then you have here, if you click on P10, you get all the mutations that have been reported with different phenotypes in P10. So basically, you are understanding right now whether the gene is a pathogenic gene or has been known to be contributing to disease mechanism. So that's the genomic content part. And then the second thing is, here we have the SV population frequency, control population frequency. So we see if this SV has been reported in the control population and reported to be harmless or benign. And then here we overlap the SV with Horizon's proprietary database, where we have a lot of pathogenic structural variants. And then we overlap with them and we check where have they been reported before, as you can see here in the PubMed ID on the side. And then we have also additional things like Horizon's pharmacogenetics database, which I'll discuss later in more detail. And there's also, again, the ability to integrate your own population-specific custom variant frequency database. Finally, again, you have also a specific cancer variant database that is both in the SMV and in the SV to see cancer-specific variants that have been reported in different cancers, as well as the inheritance type, whether it's somatic or germline or otherwise. So then all of this has been how can we interpret the variants and prioritize them. Then if we look at the types of variants in terms of inheritance, we see that we need to sometimes analyze the proband by themselves, and sometimes we need to analyze the family plus the proband. And when we analyze the family plus the proband, we can identify de novo variants and inherited variants. So in this case, this is a singleton analysis, this is a trio analysis, and they're both very important. And so Horizon helps with that through the different modes of analysis it provides. So when you're talking about doing this manually, you need to imagine that you'll be opening a VCF file of 5 million variants. You'll be overlapping it with another VCF file of another 5 million variants, and then you'll be checking which variants are overlapping, which variants are unique to the proband, and then you can start doing the interpretation process. So it's very hectic, and we don't just do that for SMVs, we do that for SVs as well. So in this case, automating the process here through trio analysis module, you can add the proband, the father, the mother, and you can specify a read depth filter, and then it will immediately show you the inheritance for each variant plus the pathogen. So you can filter out for de novo variants. You can check for paternally inherited variants, maternally inherited variants. This even helps with identifying compound heads easier. So I'll be showing a case study on that after this as well. So then the fourth thing here is not just looking at, so right now, all of what we've been talking about has been mostly directed or focused on the primary analysis, which is we have a phenotype or a disease, and we're looking specifically at that. Or it might be an unbiased look. However, if we imagine that all of this, if we think that, like, if we consider that all of this we've been looking at the primary phenotype, then this step is more about what can we accidentally find or what can we incidentally find within the patient that will determine not just the present state, but the potential future state of the patient. So there are two things that Horizon can help with. Number one is the carrier screening, and number two is ACMG secondary findings. So for carrier screening is basically finding variants that are that cause the patient to have a carrier status. Basically, they can develop the disease. They can let's restart this one. So carrier screening here is finding variants in a proband that can later cause a disease in the offspring when they combine with another patient. So in that case, it will identify what disease might arise in the future due to the risk assessment of certain damaging variants. So Horizon right now can screen up to 500 genetic conditions, including cystic fibrosis, Fragile X, retinitis pigmentosa. And what you do is that it's simply, you can choose the carrier screening workflow and then analyze, and it will show you here the carrier screening information for each variant. So this is a variant where it shows you the disorder related to it, and then it shows you the inheritance here, autosomal recessive, and then the gene name. So now you can understand that this patient is heterozygous for this variant. But then if they later get married and they are married to somebody who has another heterozygous form or homozygous form of the variant, then it's very likely that then it's likely that the offspring might develop this disease. So this is very important for genetic counseling. And again, it gives us a sneak peek on the future of the patient. And then the next thing is ACMG secondary findings. So ACMG secondary findings, ACMG has published a list of secondary findings genes that are actionable findings for different diseases that are unrelated to the primary purpose of the analysis. So secondary findings is basically So then the second thing is ACMG secondary findings. So we have a list of genes that have been published by the ACMG, by the American College of Medical Genetics, that need to be reported. And secondary findings are basically actionable findings for different diseases that are unrelated to the primary purpose of the analysis. So these genes are about 80 something genes, and they are actionable and should be reported. For example, one of them is P53, where you have it's related to cancer. So then, for example, the patient has come for kidney problems, but then you find P53, so then you need to report it because they might have cancer. And then finally, pharmacogenomics and multi-omics integration. So now we've talked about connecting phenotype to genotype. We've talked about analyzing SNVs and SVs. And then we move to family and singleton and trio analysis, where we look at inherited versus de novo variants. And then we screen for additional findings like carrier screening and ACMG secondary findings. And now we look at how can we integrate this genomic data into other omics. So one of the main things, of course, is pharmacogenomics. So Horizon utilizes a manually curated pharmacogenetics database with over 1,000 genes associated with pharmacogenetics and drugs. So for example, in this here, you can see that this is a structural variant that is impacting multiple genes. But then you look here, and you see that for each gene, Horizon has identified a drug that is actually connected to this gene. And then you can see here on the side the PubMed ID leading towards this link. So this is crucial in finding potential adverse drug reactions in patients before moving forward with a treatment plan. It allows clinicians to design therapeutics and personalized treatment options, and then this helps in therapeutic intervention management. So if we look at the entire picture, we see that we have seen the genomic profile of the patient right now. We have glimpsed the potential future the patient might have or might develop, and then what we need to monitor, and then even how we can change or improve the treatment management based on these gene drug links. And then, of course, all of that, we can just automate it, and we get a final report. So we get the patient basic details and then the variant information for SMVs and SVs. And then you can add here some comments on the interpretation itself. So an automated report that can immediately go. Now, all of this, how long has it taken? Well, the automation process has reduced this from 24 hours plus into 20 minutes and 5 minutes. So you can see, just to summarize, how a lot of things, a lot of factors go into streamlining the process, and how through the integration of AI, through the integration of large language model, through proprietary databases, integrated analysis, you can see that all of this is automated and provides you a report, provides you integration with other omics, and enables fast diagnosis in minutes. So let's see how this can be used through actual case studies. This is a rare disease case. Here we can see the case when we have a de novo variant. So this variant has been identified in a patient. This is an example variant. Here we have in chromosome 12, KMT2D. This is a de novo variant. It was identified here through the trio analysis by Horizon. And what we did is we added the proband, the father, the mother to the trio analysis module. And then the inheritance here was de novo. The variant was classified as likely pathogenic. And then we confirmed the de novo status of the variant through IGV. So you can see here, this is the proband reads, this is the father, and this is the mother, and this is in the KMT2D gene. You can see here that the variant is only in the proband, and it's not found in the mother and father. So this is very important, and this adds to the evidence that the variant might be pathogenic or likely pathogenic. And then this is another case. It's very interesting. It's a compound heterozygous case where, again, there are two variants that have been identified by Horizon. You can see that the positions are very close to each other, and then they are likely pathogenic. And then we wanted to verify that, so we again looked at IGV. You can see here, this is the proband, this is the father and the mother. So you can see that the proband, this is the first variant, and the second one, they're both in HBA1 gene. You can see here that this variant here is from the father, while this variant here is from the mother. And then you can see that they are not overlapping in the reads, so you can see that when there is a shift here, a mutation here, it's not here on this read. So that means that they're on different alleles. So together they are compound heterozygous. And this is very important to identify. These are complex cases that manually would be very hard to find. So then it's very important to understand the accuracy behind this, or what forms the basis of this interpretation. So we did a validation study on the copy number variation. Currently, we did a cohort with different phenotypes, including NDD. And then we did ROC analysis, receiver operating curve, characteristic curve, which showed an overall accuracy of 96%. So the paper is actually right now in bioRxiv. We're in the process of publishing it. So you can see here the draft. And then here, what we did is we evaluated the total accuracy across different cohorts of Horizon's CMV module. And then we found 96% overall accuracy. And then the second thing is we evaluated CMV accuracy across deletion and duplication CMVs. So you can see here in this graph, we have a slightly higher accuracy in duplications versus deletions. And then we evaluated the accuracy also across different CMV sizes. So you can see here, these are different sizes, less than 1 MB, 1 to 3 MB, 3 to 5, and above 5. You can see all of them are above 0.9, above 90% accuracy. So it's really amazing, and it can really have a huge impact on the patient management and also in genomics and research. So now this is the fun part. Let me show you a sneak peek of Horizon version 2. So the next version is going to be amazing, even more amazing than now. So it's going to have a multi-omics update. We're going to have different omics that will be integrated into Horizon. There's going to be a huge upgrades as well in the analysis and interpretation part. So stay tuned. There will be an early access program coming soon in October. And we'll be launching the registration for that. So we'd love for you to try it on your data or on any data. And it will really integrate with different omics. It will really transform the platform from just genetic interpretation to a multi-omics platform. So thank you very much for listening. And if you have any questions, please don't hesitate to email me. It was a pleasure today to discuss this.
Amazing talk, Mariam. It's really very insightful how Horizon can solve many issues arose from this complexity of rare diseases. And I would like to know certain questions that actually came from different scientists and clinicians. Hope you can, enlighten us from your experience of using Horizon platform. So one question is how GenomeArc Horizon will impact tertiary analysis for NICU, PICU, and when we are racing against time for diagnosis, how GenomeArc Horizon can create a huge impact.
Yes, so actually, for NICU, PICU patients, as we discussed, the most important matter is timing. So then, what Horizon does as the greatest value, in my opinion, is how it automates the bottleneck of genomics. Because the main factor that you need to consider when you're talking about critical case patients is how to get accurate, efficient diagnosis in time, in very, very fast time. So I think definitely GenomeArc Horizon platform can help with this and can speed up the process and can save a lot of lives.
Second question, can GenomeArc Horizon predict pathogenicity for all types of variants in a seamless manner, not using different types of tools, but in one platform? And is it compatible with the new, long read technology that we have? Is it compatible with long read genome VCFs? Because those genome VCFs have different structural variation formats. Is it compatible with that?
Yes, so it's actually compatible with all kinds of variants. It's all in one unified platform. If you look here, sorry, if you look here at this part, you can see that there are three modes of analysis, and you can just upload here the SNVs, and you can upload here the SVs, and you can even use that to identify de novo mutations. You can use that. There's another module that I haven't went into depth with because of the timing. It's the tumor one where it identifies somatic mutations. For cancer patients. So it really has a lot of capabilities and it all happens in one unified platform. So you can upload all these variants in the same platform, one file for SMVs, one file for SVs, and then it will automate the process and interpret all of them. Also, for long read and short read, yes, it can be used for both. It's actually been tested with some long read files and it's worked with different sequencing machines. So it's compatible with long read, short read, and it looks at germline and identifies somatic variants as well.
So the next question is, can you use custom workflow and add, let's say, custom population databases with frequencies so you can filter out population specific variants or common variants in that population? Does GenomeArc Horizon platform allow that?
Yes, so here for the population specific variants or frequency, if you see here in the SNVs, there is a third type of database that can be queried by Horizon. So this is the custom databases uploaded by the user themselves. And what it does is that basically if you have a specific population uploaded by the user, Horizon considers that when it interprets the variants and checks the cutoff that you mentioned. So it does actually have filters for population specific. It also has an additional filter for read depth. If you see here, you can, this is for total read depth. So you can set that and it will filter out all the variants that fall below the cutoff that you specified.
Thank you, Mariam. Can the language model, Horizon Intelligence, the language model predict disease and create gene panel for, to aid diagnosis? Can it also integrate this outcome with genomic data, especially the genome variant data? We just want to know from unstructured clinical data integrating with the VCF, can you design a panel and identify a variant of interest linking the phenotype with genotype?
So for the NLP, it's really amazing. I haven't seen a lot of things. I haven't seen like it integrated before in the tertiary analysis. So what it does is that it takes this is an unstructured clinical phenotype. It's a text description in our language, just basic language, basic description of the symptoms that are shown by the patient. Now what it does is that it takes this, it transforms it into structured data, and then it outputs, as I mentioned, three kinds. Number one is the HPO terms, number two is the genes, and number three is the diseases. So it shows you from this basic description, from the unstructured data, it provides you structured data sets. It provides you HPO terms, it provides you probable diseases, causal diseases, and it provides you causal genes. And this is amazing because, as I said, it has a huge effect on diagnostic odyssey. It can help also in the interpretation with curating a gene panel, and it really shortcuts the time.
Thank you. So this will be our last question for the webinar. So we'd like to know if GenomeArc Horizon platform, integrates multiomics, and if it does, what kind of multiomics it integrates, and how it helps the diagnosis and therapeutics of rare diseases.
So for multiomics, currently we have pharmacogenomics. You can see here. For each gene, we output the pharmacogenetic information, the drug links, so the gene drug links, and then all the data that supports this. And then there's also transcriptomics. So if you look here, you can see this is transcriptomics. Where is the gene most expressed in which tissue? That's also very important, because if you're looking for a specific disease, you would like genes that are expressed maybe. If you're looking for an NDD, neurodevelopmental disorder, you would like to see genes that are expressed mostly in the brain. Or it could bring you different insights to see which genes are expressed where. So this is transcriptomics. So we have transcriptomics, we have pharmacogenomics, and then there are, as I mentioned, there are more omics coming in the next update. So it will be very exciting. We will have single cell omics. One second. So this will be in the new version. There will be multiple updates, especially in the multi-omics part. So stay tuned for that. And there will also be an early registration, as I mentioned. You can sign up for that and try it before it's launched officially. Thank you very much.
Thank you, Mariam. It was a nice session. I hope everybody enjoyed as much as we did, and we look forward to have more of these clinical genetics, webinar hosted by GenomeArc, and we will invite all of you again. So see you soon. Bye.